Let me introduce Professor Shailesh Singh sir. So Professor Shailesh Singh sir obtained his PhD in cancer biology from Institute of Medical Sciences, Banaras Hindu University. He has been a scientist in the James Graham Brown Cancer Center, University of Louisville, School of Medicine, Louisville, USA. He is currently working as a professor, Morehouse School of Medicine, Atlanta, GA, USA. He is also a member of Cell and Molecular Biology Program, Winship Cancer Institute, Emory University School of Medicine, USA. He has been teaching medical students and is course director of Cancer Biology and Integrated Biomedical Science Program. His area of interest is development of therapeutic and preventive strategies using immunomodulatory natural compounds. He has more than 100 publications and over 60 patents. I mean, quite a remarkable achievement and accomplishment. He has been recipients of several awards, the latest one being Outstanding Scientist Award, Association of American Asian Scientists in Cancer Research 2019. We take this opportunity to congratulate sir for such an achievement, although this has been done prior to the COVID. Professor Singh is a great mentor of students. I think you are all very fortunate, although many of you are already faculty members. I think you will be benefited by his interaction in future. And he has mentored more than 4 dozen students of MD, which is remarkable. I mean, people from the medicine, and then MS, surgery, a PhD, and a postdoctoral fellow. He's a great philosophical orator of life in perspective of science and karma for divine pleasure. So with these few words, I now hand over the microphone to Shailesh sir and request him to start his lecture. And he can also start with a very general lecture before going to the main topic, if you want to share something outside of this in the beginning. Thank you, sir. Welcome, sir.
Yeah, thank you. Thank you so much, Dr. Bora, for a nice introduction. And I will be very brief. And you know that, as you said, that this there is a diverse people attending this course. So I tried to make it as simple as possible. And as I mentioned, that after teaching in United States from 21 years, the students are very different here. So I have to be very brief, and I will be just talking about DNA breakage. Okay, so what may cause it, and what are the repair mechanism, why the repair should happen, and where the repair is important and where repair is not important, because every time we do not want the DNA to be repaired, because in a clinical perspective, sometimes we do not want DNA to be repaired, because otherwise some of the therapeutics doesn't work. So I will be going briefly overall, okay? So you all heard since morning, and Dr. Bora also presented this picture, okay? So so first of all, I prepared a basic slide of the DNA, and you all know, and I'm not talking about aDNA, bDNA, and zDNA, okay? So because it is just, I'm talking about the natural form of the DNA. So you can see this is the DNA, and you can see how it is, what are the purine and pyrimidines, the basic structure, okay? So you have ATGC, these are the 4 things, which is giving the whole blueprint of life, okay? So that these are the only 4 things which is making the whole life on this planet. And any kind of alteration, any kind of damage, any kind of change in the sequence is causing the problem, okay? And it's not always the problem. I will talk later. And here, you can see how the ATGC is paired, and this is basically called that Chargaff's rule. Okay, so A should always pair with the T. Okay, and the side and C always should pair with the G, and most important thing you can see here, the bond. Okay, so between A and T there is a double bond, and between G and C there is a triple bond. And you all know that the difference between double bond and triple bond. Okay, if it is a triple bond, the bond energy will be more and bond length will be shorter. Okay, so that may be the reason you are getting a electrical structure. Okay. this is the picture like showing you the replication. A replication always happens in the 5 to 3 prime. And the reason why I'm showing replication because the error may occur during replication as well, okay? Now coming to the point, okay, DNA damage, okay? So this is what basically happens. Like, you know that DNA damage may occur naturally during replication by any insult or in environmental insult, or you insult your body by taking wrong thing, like eating wrong thing. And when that happens, okay, so to make things normal, okay, so everything should follow the law, okay, so the replication should be faithful and like genome should be maintain in its integrity, okay. So if DNA doesn't have, the damage caused is not repaired, so it is basically leading to the genetic instability, and it's good or bad, okay. So like you have to always think philosophically, because if there is no instability, basically gives the opportunity, okay. So you have to learn philosophically, like once you are stable, you don't explore, okay. So once you are unstable, you explore. So instability sometimes creates the variation, and that's the nature's law, but sometimes instability is not right, okay. So and during that repair, if the repair is not 100%, okay, sometimes the repair may be 100%, sometimes it may not be 100%. So if the repair is defective, incomplete, then the genetic stability is leading to the genetic divergence or cancer or the hereditary disease, what we see more often, okay. So what causes a DNA damage? Okay, so damage means the DNA damage is an abnormal chemical structure in DNA. So anything which is causing an abnormality in the DNA structure is considered as a damage. Okay, an agent that damages DNA, we are exposed to like radiation, and then highly reactive oxygen radicals, okay, produced during the normal cellular respiration, okay, so that's called the oxidative damage. Ionizing radiation such as the gamma rays or the X-rays, like we are exposed to, that causes the damage. And ultraviolet rays, okay, especially the ultraviolet C, between the 200 and 260 nanometer, and because that DNA absorbed highly, okay, but also the longer wavelength of the UVB that penetrates the ozone shield. Then chemical in the environment, okay, so we consume like a lot of things like, okay, more aromatic hydrocarbon found in the basically like found in the cigarette smokes and other form of the aromatic hydrocarbons we get. The way we like cook certain things, okay, like basically when you are grilling certain things, it's not a common like diet like in India, but like, you know, the charcoal grilling is very common in the United States, and that basically causes more like damage and major contributor of the colon cancer. And the plant microbial product like aflatoxin produced by the moldy peanuts, that is causing the DNA damage. And the chemotherapeutic agents, okay, all the chemotherapeutic agents used like for treating cancer are DNA damaging, okay, because that is the mechanism for any chemotherapeutic drug, especially the platinum based drug, to work. So coming to the like what type of DNA damage is happening, okay, so like, you know that when I said that any kind of alteration in the building block of the DNA, okay, so if anything is changing, okay, so that is considered as an alteration. So it's a depurination, okay, so you have like an deamination of the cysteine to uracil, deamination of adenine to hypoxanthine, alkylation of the bases, okay, insertion or deletion of the nucleotide. So like if the nucleotide should be like a proper, if there is a insertion or the deletion, and base analog incorporation, if anything which is mimicking the base but actually is not the base, okay, so like, you know, probably the structural similarity but it's not the actual base if it is like incorporated during replication. Okay, so and other is the two base alteration. So it's basically done by the UV light, okay, induced like thymine dimer, or chain breakage that is caused by the ionizing radiation, radioactive disintegration of the backbone element, oxidative free radical formation. Then the cross linkage. The cross linkage is between the base in the same or the opposite strand. So sometimes it get cross linked, and between the DNA and the protein molecule. Okay, so there also, because you heard a lot about like histone and other things in the first lecture, and after that, like you heard from Dr. Bora about the binding proteins. So this is the picture showing you the spontaneous damage. Okay, so deamination. So you can see here in the picture, like the amine is removed. Okay, so and amine is removed. So it is not a natural like form of like of cysteine of the cysteine. Okay, and depurination, where you can see the purine is gone, and that is considered as an unnatural like a structure, and which may cause the like which is not maintaining the integrity of the DNA. Then it's the induced form when it is a UV exposure. Okay, so you can see the adjacent thymidine in DNA, and you can see that like a thymine dimer here. Okay, so when the dimer is formed, as I said, dimerization. So this dimerization is considered as a like not the natural integrity. And then alkylation, okay, when the guanine is alkylated. So this is the basically like the complete take home. Okay, so this is the summary of everything which you should at least like carry with you. Okay, so importance. What is the importance of DNA damage and the repair? Okay, so as I said, like these are the damaging agents. Okay, X-rays, oxygen radicals, alkylating agent, spontaneous reactions. UV, okay, polycyclic aromatic hydrocarbon, X-rays, and the chemotherapeutic agent, and any kind of replication error. Okay. So what it's causing, like when it is X-ray, X-rays, oxygen radicals, alkylating agent, or spontaneous like reactions, they basically cause the single strand break. Okay. If it is a UV, polycyclic aromatic hydrocarbons, then it is causing the adduct. Okay. And if it is a X-ray and the chemotherapy, it's the interstrand cross-linking or the double strand break. Okay. During replication error, it will be mismatch, okay, or insertion or the deletion. How it is corrected, okay, is like if it is a single strand break, it's a base excision repair will occur. If it is adduct formation, it's a nucleotide excision repair occurs. Okay, so that is the mechanism like you use to repair that problem. If it is an interstrand crosslink or double-stranded break, then recombinational repair happen. Okay, so and if it is a mismatch, which happens mostly like during replication, then the mismatch repair should occur. So what happened? What is the consequence after that? If this happens, how the biological system is taking care of it? Okay, like you know that of the if it is corrected, it's all right. If it is not, then first thing will happen, the cell cycle arrest, because in the body everything is kind of tightly regulated. It's like you know that even if you go for the detail in the DNA replication, there is a lot of proofreading. Okay, and if anything fails, still there is a way to take care of it, because every abnormal cell has no existence in the body, and if anything is trying to overcome that, okay, because a biological system will try to fix or eliminate anything abnormal. If somehow the cell which is carrying abnormal DNA or the damaged DNA is not corrected, then the first thing may happen is cell cycle arrest. Okay, so the cell will be arrested so that it is not making another copy. Second consequence may happen, like it will the inhibition of transcription or the chromosome. And if that happens, cell will go under the apoptosis. So there is a big mechanism of apoptotic cell death. Okay, so the whole point is that it's another check and balance, that anything which is abnormal or not following the law of the nature's law should be eliminated. Okay, then mutation or the chromosomal like aberration. So, and that basically, if that happens, okay, so that's the serious condition. Then cancer, aging, and there are a lot of inborn diseases occurs. Okay, when the cell is not going to die, then, and if it is escaping the cell cycle arrest and cell death, because in case of cancer, you all know that if there is a cell cycle arrest is a big issue, because if the cell cycle is, if the cell is not proliferating, and this and there is a altered DNA in that cell, that causes the more like a fatal condition, because more often that contribute to cancer. Okay, and that is non-treatable, and that is the reason for relapse, because more often when you try to like kill a cell with a different like therapy, the first thing cell does, because go in cell cycle arrest. Okay, so because every treatment or every clinical like that intervention we have as of today only go for the proliferating cell. Okay, second point, when you are talking about the cell death, if a cell which is a defective or damaged, or it has a mismatch, like mismatching, insertion, deletion, or adduct, if it is not dying, it is creating a like it is creating a fatal condition. And you all know that the first thing a cancer cell does, avoid cell death. Okay, and increase the increase their ability to survive. So the survival will be higher, death will be low. Okay, so let's go on one by one, like the mechanism of repair first. Okay, so how this like all repair mechanism takes place. Okay, so coming to the repair, like a cell have a multiple repair system. Okay, so like you know that reason why it has a multiple repair system, because you know that so that take care of the multiple insult. Okay, so like if the cell is not, so there's it is giving it a more option to take care of various type of damages. Okay, so repair is possible largely because the DNA molecule consists of two complementary strands. So the reason why everything in nature has happened for a reason, because if there is only like one strand of the DNA, and if it is damaged, there is no reference. Okay, so the nature has created the reference, and it has given you the two strand of the DNA. So a reference like a DNA can be used at least to fix the issue, so that like system is reading what is missing. So if suppose like if we have just one and there is a T missing, okay, instead of T there was a G inserted, how the system is going to know what was real? So it's basically giving you the reference. Okay, so having a multiple DNA repair system gives the opportunity to fix the various type of damages. Okay, and repair is possible largely because the DNA molecule consists of two complementary strands. So coming to the mismatch DNA repair. Okay, so you know that you can see here like this chart, okay, during the replication. Okay, so you can see this is the replication, and for a short period of like following replication, the template strand is methylated and new strand is not. So you know what's the important of methylation? It's a tagging. Okay, because you know that if how a system knows, we look at the figure and we always say, okay, it's a parent like a strand, and it's a new strand. But somebody is showing us, because we see the picture, how the biological system is recognizing. The way biological system recognize something, that you're always the parent strand is methylated. So so like the system is knowing the one which is non-methylated is the new one, and any problem happens, I have to read the methylated one to fix the problem of the non-methylated one. So, after a few minutes, a new strand is methylated and the two strands can no longer be distinguished. So if the both strands are methylated together, biological system is not going to figure it out which one is original, which one is copy. So methylation is important to identify the original and the non-methylated one to identify which one is becoming. So you see, like if the both are methylated now, no way the biological system can fix this issue because your reference is gone and you don't know, like it's a kind of that you have a like a multiple copy. Okay, so like if you make a photocopy so original that you may not be able to distinguish which one is old, which one is new. So coming to here, like the way that it is any kind of damage is fixed in the mismatch. You see the mute S and mute L, it basically comes and forms the complex. Okay, and it's the ATP dependent, like energy dependent process. Then mute H comes and forms the complex where there is a like mismatch. Okay, and then mute H cleaves and unmodified strand. Okay, coming to the how the mismatch is completed. Now you see that it is just a continuation of the picture, because I thought of making it bigger other than making it smaller so that it's visible. So mute S, mute L, and mute H. Okay, so it's like it basically coming, and you can see here like it is using that DNA helicase 2 exonuclease or RECJ nuclease, and then DNA polymerase 3 is basically fixing here the single SSB. Okay, and so next I showed in the picture is the base excision repair. Okay, so like you can see in this picture here, so there is a problem with the base. Okay, so the DNA glycosylase recognize the damaged base and cleavage between the base and the deoxyribose in the backbone. So this is the wrong one. Okay, and this is a damaged base, and then AP endonuclease cleave the phosphodiester backbone of the AP site. So you can see the phosphodiester is broken here. And then DNA polymerase 1 initiate the repair. Okay, so like you see the DNA polymerase 1 is initiating the repair here. And then basically that ligase, as the name says, that ligating something is ligating and fixing the issue. Okay, so that is how the base excision repair is performed. So coming to the nucleotide excision repair, okay, DNA lesion that cause the large distortion in the helical structure of the DNA generally are repaired by the nucleotide excision system, and the repair pathway critical to the survival of all free living organisms. And coming to, let me move these things like it is coming always on top of. Okay. 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 So you can see here, like, that the lesion, you can see this DNA lesion region. Okay. So the first thing happened, cleaves the DNA damage, stand on the side, either of the lesion. And the second step is DNA is removed with the aid of the helicase. So the enzyme which is used is the helicase, which is removing it. And then gap is filled by the DNA polymerase. In case of like prokaryote, it's a polymerase 1, and you all know that in eukaryote, like it's not a polymerase 1, 2, and 3. It's a holoenzyme. So the part of the polymerase which is playing significant role here is the DNA polymerase epsilon, and then the remaining nick is sealed by the DNA ligase. So the DNA ligase is coming and fixing the problem. Then coming to the direct repair, so these were the repair where all this machineries were involved, and several type of damage that are repaired without removing the base of the nucleus called the photo reactivation, okay, of the cyclobutane pyrimidine dimer, which is basically caused by the UV damage and the reaction and reaction promoted by the DNA photolysase, okay. The pyrimidine dimer result from an ultraviolet light, okay, and induce like reaction and the photolysis use the energy derived from the absorbed light to reverse the damage. So it's basically that same thing which is damaging, the same thing which is activating, but at the different like wavelength. Okay, so this is the picture like showing how the pyrimidine dimer is fixed and by photo reactivation. Okay, so the first one you can see this is the like light coming, and the blue light, a photon 300 to 500 nanometer wavelength is absorbed by the MTHF polyglue, okay, which function as a which is functioning as a photo antenna. So damage is basically caused. I showed you at the 260, but for repair to happen, the wavelength is now that 300 to 500. Okay, and you can see here the excitation of energy passed to the FADH into the active site of the enzyme, and then the third here you can see that excited flavine donates an electron to the pyrimidine dimer, okay, to generate the unstable dimer radicals. Then coming to the fourth picture, picture, you can see the electron rearrangement restore the monomeric pyrimidine. And coming to the fifth one, it's the electron. The electron is transferred back to the flavine radical to generate the FADH. And now you can see the damage which was caused is now fixed. So here there is nothing going out. There is nothing going out. There is no there is nothing cut. There is nothing removed. There is nothing inserted. It's the same like a molecule which was damaged was repaired without any excision. Okay, so other method when we said is like a recombination, where we said where we said that there will be like the chemotherapy and others are causing the damage. Okay, so the rearrangement of genetic information within and among the DNA molecules. And so recombination is a big method, and it is not only for fixing the issue. It happens during cellular proliferation, but I'm just talking about the homologous genetic recombination is a primarily a DNA repair process. So out of recombination, homologous genetic recombination is a DNA repair process. And this is the homologous, this is the recombination process. Okay, so you can see the double stranded break in one or two homologous is converted to the double stranded gap by axon of exonuclease, and strand 3 are degraded less than those of the 5, okay, and producing the 3' single strand excision. Okay, so you can see here, and then an exposed 3' and pair with the component in the intact homologous other strand of the duplex displaced, and then third is basically coming here. Okay, so because I cut the picture. Okay, so so invading 3' at the is extended by the DNA polymerase plus the branch migration, eventually generating a DNA molecule with the 2 crossover of the Holly intermediates. And further, the DNA replication replace DNA missing from the site of the original double stranded break, and then you can see the cleavage of the cleavage by specialized nuclease generate either 2 recombination product and set 2 and the DNA on the either side of the region undergoing the repair. So you can see the product 1 and product 2. So what is the consequence? And this is basically the summary of what I just said: the source, consequence, and clinical outcome of the DNA damage. So what is the benefit and what happened? Okay, so Incentric damaging agent and eccentric. So anything which is coming within the body and anything which is outside the body. So within the body, the damage cause is like the reactive ROS, replication mistake, replication stress, alkylating chemicals, metabolically derived aldehydes. Okay, so this is the the summary slides, 2 slides what I showed. If you remember that, I think the purpose is solved. Okay, and eccentric is the radiation, environmental, chemical, and the chemotherapeutic agent. Okay, so the damage caused. Okay, so it's active damage, idle damage. Okay, so within the idle damage, it's a mismatch, base lesion, bulky adduct formation. I talked in active damage. Okay, SSB and DSP. Okay, single strand break and double strand break. What is the molecular effect? It's a mutagenesis. Okay, so these two cause the mutation. When there is adduct, it's a block transcription, block replication, mutagenic bypass. Okay, when it is a single strand break, so it's a block the transcription, faithful recombination. If it is a double strand break, then block transcription, replication for collapse, genome, it contributes to genomic instability. What is the cellular consequence? Okay, if this happens, if the mismatch is a transformation, if it is base lesion transformation, bulky adduct transformation, and the cell death. If it is a single strand break, it's a cell death. If it's a double strand transformation or the cell death. What if that happens, not able to fix, what happened? Okay, so it's like a disease outcome. It's a cancer plus immune dysfunction. It contributes to, when it is a adduct formation, it's a cancer, neurological disease. Okay, premature aging. So if mostly the adduct contributes to the premature aging. Or you can say when we age, we have no it's a vice versa. When you age, there is a repair mechanism weak. Okay, so that's another perspective of looking at it. Neurological disease. Okay, when it is a single strand break, and when it is a double strand, it's a cancer, neurological disease, or immune dysfunction. Okay. So we talked about what are the responsible pathways. This is just a summary. Okay, MMR, BER, NER, SSBR, and DSBR. So we just spoke like, so this is the summary of what I said. What may cause, what is the molecular effect, what is cellular consequence, what is the disease it may cause, what is the clinical consequence, and what is the repair pathways which are going to fix these problems. So if there is like a DNA repair deficiency disorders, okay, so you just heard about that. These are the some of the DNA repair genes and the proteins and their repair pathway. Okay, what are the repair pathway affected, and what happened? Okay, so you heard just the BRCA1 and BRCA2. Okay, so basically it is basically HHR of the double stranded break and daughter strand gap. And if there is the issue with that, it contributes primarily to the breast cancer and the ovarian cancer. Okay, ATM, it's a different mutation in ATM produced HRR SAA, and it is contributing the leukemia, lymphoma, or the breast cancer. Okay, NBS is contributing the lymphoid cancer, and MER11 is contributing to the breast cancer. So these are basically the gene where there is a no a repair done. They may contribute to WRN, contribute to soft tissue sarcoma, colorectal, skin, or thyroid or pancreatic cancer. RECQ4, it is causing the basal cell carcinoma, squamous cell carcinoma, or intraepidermal carcinoma. And these are some more like FANCA. It basically causes that leukemia, liver cancer, solid tumor of the many areas. Okay, XPC, XPE is contributing to the skin cancer, and these XPA, XDP, XPG, XPF is contributing to the skin cancer. Okay, and XPV is contributing to skin cancer, basal cell or squamous cell melanoma. and MSH2, MSH6, MLH1, and the PMS2 is basically leading to the colorectal or the endometrial cancer. And you all know this is more studied. Like if there is an issue with the P53s, it is leading to the like sarcoma, the breast cancer, brain tumor, and anti-HL1 is colorectal cancer, endometrial cancer, duodenal cancer, basal cell carcinoma. So if there is a DNA repair deficiencies, like this is what you get. And then, like as I said, like always, the repair is repair. Sometimes we need repair, sometimes we do not need repair. So most of the time, you know that because the DNA repair is causing chemoresistance. Okay, so like all the treatment we provide, if the cell is learning how to repair, then it's a problem. And what happens? The cell cell tries to overcome the therapeutic intervention by upregulating the repair mechanism. So any kind of damage you cause, they will repair. So you can see here like the DNA damage response. What happened to the DNA damage response? So like oncogenesis, transcription factor, transcription factor, cancer stem cell. Okay, bystander signaling, hypoxic cancer cell. Okay, so you know that it's a like when so what happened, like when you are giving the chemotherapy, so you can see like what you suspect when you give any kind of chemotherapeutic agent. As I showed before, it should cause a damage. Okay, like either it's a single strand or the double strand. Okay, and that accumulation of adduct basically push cell to die. But the cancer cell, they learn how to repair this so quickly, and that is basically contributing to the chemoresistance. So that's it. And if you have any question, I will be more than happy to answer.
Shailesh sir, you can start your video if it is possible.
How it got, okay. Yeah.
Yeah. Thank you.
Yeah, I was very brief. I just want them to know what is damage.
It was an excellent lecture, and
what is damage and what causes damage and what is the clinical significance of damage, and when the damage is good, when damage is bad.
Yes, sir. Yeah, so kindly send your PowerPoints to us. So
I will email you. I will email you the PowerPoint.
So there are some questions. So participants, you may start asking. So there was a question. What does lesion means? Probably we are speaking about DNA lesions by Sonali Kelkar. Yes, sir. What is a lesion? She is asking.
Let me see.
Yeah, can you see the set box? Yeah. Somebody is writing, sir, how does P53 sense DNA damage?
P53 is not sensing the P53 is not sensing the damage. Okay, the P53 is basically like anti-oncogene. Okay, so like that if there is like any kind of damage like a cause, the P53 may not allow it to get fixed. So it was studied as an oncogene before for like 10-15 years, and then we started like studying that it is an anti-oncogene. So it is basically like any kind of like irregularity. It is maintaining the integrity of the DNA. And if there is a problem with the P53, then any kind of problem like P53 and especially the P10, because the P10 is the same thing controlling the PI3K and AKT. And if there is a mutation in like P10, or the P10 is abnormal, then that may contribute to the cancer progression. Because in everything we know, like in a biological system, there is nothing which cancer is using specifically. and all the molecule, all the pathway, all the damage, all the cell cycle checkpoints are required for a normal functioning. Only thing is that it's a dysregulated, and cancer is using it for its advantage, and strategically, like point by point, like that sometime like it may upregulate something, sometime it may downregulate something.
Yes, sir. So there is any other question? Does DNA damage by various agents activate tumor suppressor factors? Does DNA damage by various agents activate tumor suppressor factors?
Yeah, because that it is. Otherwise, like there is a complete possibility, because that either like either you can it's a either you accelerate or press the brake. So that if there is no brake, then the chances of accident is more. So there is absolute possibility that they are altering that. Either you promote, either there is a promotion, or there is the defect in something which is controlling. So yes, the possibility. So any kind of external like mutagen, or any kind of external like product we are eating or consuming, that may contribute to the cancer by altering the anti-oncogenes.
Yes. So there is another question. Komal Taneja asking, sir, you mentioned in some cases DNA damage is advantageous. Could you please explain it with an example? I think you did it actually for chemotherapy, but so you can
Yeah,
be
because
a little bit more elaborate on this.
the whole purpose of the chemotherapy is that we are like giving all these like intercalating agent like cisplatin, carboplatin to cause adduct. Okay, in normal course, like we have to fix the adduct, but the reason this the by forming the adduct, we are promoting cell death. Okay, so like we are eliminating cancer, but if the damage is repaired, then it is compromising the chemotherapy. So at that point, that if our intention is to cause damage, and when we are intended to cause damage, it should not be repaired. If the cell learn how to repair it, then the purpose of chemotherapy is compromised.
Yes.
Regular
Yes.
damage is not good, because regular damage may cause cancer, but when we are treating cancer, we want that damage to be there and should not be repaired. So that's why that when you are coming with a lot of combination therapy and other things, that's why it is very important for understanding the fundamental like biochemistry, that what are the molecule which are fixing that, what are the protein fixing that, so that you can just like come with a combination, developing something to block those proteins, so that DNA, so that repair is not happening at that point. Other problem we encounter the systemic that infusion. So that when we develop something, we do not want like a damage to not to be repaired in whole body. So that is where it's like challenging, because in an in vitro system everything looks so controlled. But if we develop something like a targeted, where we can just target our like that particular cell or the particular area, I think that is the one of the like most that is one of the challenge, like the reason why platinum drugs are not, they are awesome, is still used, but they are not so efficacious because of the systemic toxicity. And like if we learn how to block those like enzymes or the proteins which is repairing the DNA in a specifically in the cancer cell, then we can do the wonder. And that all this chemotherapy we will be getting 100 or 200 fold less, and if still getting the same clinical response.
Yes, sir. So there is another question. Are epigenetics by Sindhuja, are epigenetic changes in a stem cell is also a DNA damage?
When you
Epigenetic changes of DNA.
when you talk about like epigenetics, okay, so like that anyway, like that most of the things are epigenetic. There is a very less genetic, like genetics. If I speak in terms of like my research focus, because everything is happening, like all cancer is contributed 5% to
Yes.
by genetics and 95% to 90% by epigenetics. So you know that any kind of insult may contribute to the epigenetic modifications, like no doubt about it. And you know that that's the major challenge, like when we are dealing with the big data or the DNA sequencing, like in a clinical perspective, even the footprint is changing by surgery. So you know that when you are putting a knife and cutting a cell, and then you are taking the DNA or RNA from there and doing the deep sequencing. What you are sequencing, the cell was trying to rescue from the death. That is the footprint you are saying, or you are looking at that something else. So that is where that the biggest challenge of like doing that, like a deep sequencing and looking at the epigenetic modification or the genetic modification, because we don't know that it is a cause or the effect. So because you know that everybody who is standing close to the body is not a killer. Probably the person was trying to rescue and somebody died. So if you see a gene or the protein with some, because you see that people say, okay, stage 2 carcinoma, I found this particular gene, but it is like hard to predict what it was actually doing in that system. So the short answer is that yes, like it is like epigenetics changes, like because is contributors.
Yes. There are no any questions, sir. Okay. Yeah. So these are all appreciations. Okay. So there is one question: is it possible in vivo or only in vitro? But what is possible? He is not writing. TV Sridharan, can you? What's your full question? You are writing: is it possible in vivo or only in vitro, sir? But what is possible? We don't know what with reference to what. Anyway, somebody is writing, sir, does dehydration of cells cause any damage, breakage, mismatch in DNA? He's giving three options: damage, breakage, mismatch in DNA, since it is necessary to maintain hydration cell for DNA helix.
Yes, because you know that it is causing damage, or it is going to cause damage or not. But if the dehydration is going to change the pH, then certainly there will be a problem. Okay, so like probably like that the change in the pH may destabilize the DNA, but it is going to cause damage, or I don't think so. But certainly the biology will be very different, because like you know that dehydration may change the pH. And you know that you heard like I think you just spoke about the in that cellular environment, and how the
Yes.
cellular environment is going to change all those like electrostatic forces, and all
Yes.
those like you know that uh, that
A to B to Z
association with a
from.
different protein. Okay,
Yes. Yes.
so
Yes.
you know that if the dehydration is too high, that whole cellular structure is collapsing. But I don't think that dehydration is going to lead to that any kind of like damage or the mutation. But it may change the cell genomic function of the DNA.
Yes. Yes. So, TV Sridharan actually is asking, is DNA repair possible in vitro or in vivo, sir? DNA repair is possible in vitro or in vivo?
If Sridharan can put all the things in vitro system, it is possible. Okay, because you know that if you can maintain every condition which is available, if you understand that what is the like a pH, and what are the enzyme, and what are the things needed, because you know that the only thing is that you know in biology the induction and environment is very important. Okay, so like if you can take a human cell and properly induce, you can make embryonic cell, then anything in vitro is possible. But you have to create the whole environment required for that particular function or the purpose in vitro system. And that was the reason we were able to clone Dali, Mali, and Pali from the somatic cell. Otherwise, you cannot make any whole like organism from a somatic cell. So if you can create it's not that control. It's hard to control in an in vitro system, but if you can control that, I am not ignoring the possibility.
Yeah. Thank you, sir. There are no any questions any further. So I now take this.
I think I was done like in 30 minutes.
Sir?
I was done in 30 minutes.
Yeah, no. It's almost a hour, including the discussion. Yes, sir. So and anyway, the participants has a very tough day. We started around 10.
Because it was long day for them.
Yeah, because we had some inauguration sessions once here and over there. Anyway, so it was a long day, and I can see.
I'm going to email you the PowerPoint and that you can. Yeah, I think I will put my email address there, which was not there. I noticed. So if
Yes,
any
sir.
student they want to contact me later, of if they need anything, I will be more than happy to talk.
Yes, sir. In the website, sir, the website we created, your email is there. But anyway, kindly put your email. In your presentation, you can put your complete communication address, and it will be fantastic. Somebody's trying to write something, Sridhar. Anyway, okay.
What
So I
is
can,
writing?
sir, read some of the comments coming from the participants. They are appreciating all the sessions were awesome. Thanks a lot. Thank you for the such pictorial explanation, and Bhanupriya writing. Thank you, sir. Great explanation. Shailendra Singh writing. Thank you, sir, for your wonderful session. And as I'm very happy that people are, I think, very much benefited by your talk. So with this, sir, I take this opportunity to thank you. It's very early morning, and I think you got up.
Thank you. So no, I will now go to sleep, because the agenda said for 3:15. Then second agenda I got, it says 4:15 a.m. and I was, since I never provided you my recording, I was just sitting to
Sorry, sir. Actually, what happened? Our program has to be a little bit modified due to another program in the Delhi office.
No problem. It was really
lunch in between. Yeah, I'm extremely sorry. So
it was really my pleasure. So attending this.
So
probably
these sessions are recorded, and it will be available for future references by whoever.
And yes, I will be more than happy to participate in future.
Yes, sir. So we look forward for the next FDP in the next financial year. So TV Sridharan is writing back your response to his question. All wonderful discussions, sir. So once again, sir, thank you so much for your great lecture. Yes, sir. So we look forward to you and meet more frequently. This is the first lecture through you in the Center for the Environment and IIT Guwahati. So thank you so much, and have a nice sleep, sir.
I will be more than happy to contribute anything I can.
Thank you, sir, and have a nice sleep, sir.
Okay.
Bye, sir. Good night. And dear participants, we will meet tomorrow. So your attendance is being taken care of. This software, my student volunteers has told, attendance we can download automatically, and from the attendance we will mark your attendance. Don't worry. So before I wind up, let me tell you, all of you are probably aware that for those who are interested, there will be a test on the last day before the valedictory session in the second half. So there will be definitely objective type questions based on the lectures and the discussions over here. So as per the AICTE guidelines, those who pass in the test, they will be given some special kind of certification. And I hope you enjoyed today's lecture. And then we look forward for tomorrow's interesting sessions. And have a nice time ahead. Bye-bye for tonight. Thank you, all of you. Good night.